Chapter 10. Tumor Suppressor Genes. In this chapter, we explore a critical set of genes that help protect cells from turning cancerous. These genes work by regulating cell growth, repairing damaged DNA, and initiating apoptosis. When tumor suppressor genes are lost or inactivated, cells can begin to grow uncontrollably. Their dysfunction is a key step in the development of many cancers. Tumor suppressor genes are natural brakes within the cell. They prevent unchecked growth by monitoring the cell cycle, repairing DNA damage, and triggering cell death when necessary. Unlike oncogenes, which promote growth when activated, tumor suppressors prevent growth, and cancer arises when they are turned off. A loss-of-function mutation in one of these genes removes the cell's ability to restrain itself. This absence of regulation allows the tumor to grow. Tumor suppressor genes are broadly divided into two categories: gatekeepers and caretakers. Gatekeepers, like RB and p53, directly regulate the cell cycle and prevent inappropriate division. Caretakers, including BRCA1 and MLH1, maintain DNA integrity by repairing mutations. Both types are essential for cancer prevention, but they act at different points in the process. When either gatekeepers or caretakers are lost, the risk of cancer increases significantly. The two-hit hypothesis explains how both copies of a tumor suppressor gene must be inactivated for cancer to develop. In hereditary cancer syndromes, the first hit is inherited as a mutated gene. The second hit occurs later in life as a spontaneous mutation in a single cell. This concept was first proposed by Alfred Knudsen in his work on retinoblastoma. It provides a foundational framework for understanding inherited cancer risk. The RB gene is a classic example of a tumor suppressor. It controls the critical transition from the G1 to the S phase of the cell cycle by binding to a protein called E2F. When RB is functioning, it blocks DNA synthesis in cells that should not divide. If RB is inactivated by mutation, this checkpoint fails, allowing cells to divide uncontrollably. RB mutations are seen not only in retinoblastoma, but in several other cancer types as well. The p53 gene is often referred to as the guardian of the genome. It plays a central role in regulating DNA repair and initiating apoptosis in cells that have been damaged. p53 is activated in response to stress, such as DNA damage or oncogene activation. In over 50% of human cancers, p53 is mutated or silenced, disabling this critical defense mechanism. Its loss gives cancer cells a significant survival advantage. When activated, p53 halts the cell cycle to allow time for DNA repair. If the damage is irreparable, it triggers apoptosis, ensuring that damaged cells do not survive and divide. It exerts these effects by binding to specific gene promoters and activating the transcription of repair and death-related genes. p53 serves as a quality control system for the genome. Its activity is crucial for maintaining cellular health and preventing malignancy. The APC gene is another important tumor suppressor that regulates the WNT signaling pathway. Loss of APC leads to excessive cell proliferation, particularly in the lining of the colon. Inherited APC mutations are responsible for familial adenomatous polyposis, a condition marked by hundreds of colon polyps. These polyps, if not removed, almost always progress to colon cancer. APC mutation is a well-established early event in colorectal tumorigenesis. BRCA1 and BRCA2 are tumor suppressor genes involved in homologous recombination, a key DNA repair mechanism. Mutations in these genes significantly increase the risk of breast and ovarian cancer. Genetic testing for BRCA mutations helps guide preventive measures, such as enhanced screening or prophylactic surgery. BRCA targeted therapies, like PARP inhibitors, have shown promise in reducing cancer cell survival. These genes have become central to personalized cancer medicine. PTEN is a tumor suppressor that negatively regulates the PI3K/AKT signaling pathway, which is involved in cell survival and growth. Loss of PTEN activity results in increased cellular proliferation and reduced apoptosis. PTEN mutations are commonly found in prostate, brain, and endometrial cancers. Its inactivation contributes to aggressive cancer behavior. Targeting downstream components of the PI3K/AKT pathway is a major area of therapeutic interest. MLH1 and MSH2 are mismatch repair genes that correct errors that occur during DNA replication. When these genes are defective, small sequence errors accumulate, leading to a condition called microsatellite instability. This phenomenon is a hallmark of hereditary non-polyposis colorectal cancer, or HNPCC. Patients with HNPCC are at higher risk for colon, endometrial, and other cancers. Early detection and genetic counseling are critical for managing this syndrome. Tumor suppressor genes can be inactivated by several mechanisms. These include point mutations, insertions or deletions, epigenetic silencing via promoter methylation, and loss of heterozygosity. Any of these events can eliminate gene function and lead to cancer. Often, more than one mechanism occurs in the same gene over time. This underscores the multiple layers by which cancer disrupts normal control systems. Tumor suppressor genes must be turned off for cancer to grow, while oncogenes must be turned on. Tumor suppressors experience loss of function mutations, while oncogenes are activated by gain of function mutations. Both types of genetic changes are typically required for full cancer development. In this sense, tumor suppressors and oncogenes work in opposition but complement each other in cancer progression. Therapeutic strategies must address both aspects. Several treatment strategies focus on restoring or compensating for tumor suppressor loss. These include gene therapy to reintroduce p53, synthetic lethality approaches like using PARP inhibitors in BRCA mutant tumors, and drugs that reverse epigenetic silencing. Although directly replacing tumor suppressors remains challenging, researchers are finding ways to target the consequences of their absence. This is a promising direction in the future of cancer therapy. Tumor suppressor genes are a cornerstone of cellular defense against cancer. Their inactivation is a critical step in many malignancies. By studying how these genes work and how they fail, we can develop better diagnostic tools and treatments. Continued research is necessary to fully harness their therapeutic potential. In the fight against cancer, tumor suppressor genes remain vital allies.